It's my pleasure talking to you today about some of the results we presented at the most recent San Antonio meeting for the KEYNOTE-522 trial. The KEYNOTE-522 trial is the first prospective phase 3 trial of pembrolizumab immune checkpoint inhibitor in early triple-negative breast cancer, and pembrolizumab was given both in the neoadjuvant and in the adjuvant setting. The trial was patients with stage 2 or stage 3 newly diagnosed triple-negative breast cancer. Patients were randomized to receive either chemotherapy with paclitaxel and carboplatin for 12 weeks, followed by AC or EC for 12 weeks, with immune therapy all the way through, or with placebo all the way through. After 6 months of therapy, they underwent surgery and then continued on pembrolizumab for another 6 months or placebo. Patients were randomized 2-to-1, stratified by nodal status, by tumor size, and by the carboplatin regimen. The study had 2 primary endpoints. One was for pathological complete response, which has been shown to be linked with long-term outcome. The second primary endpoint was event-free survival assessed by the investigators. We have previously demonstrated that the trial improved significantly and in a meaningful way path CR rates from 51.2% to 64.8%. And we had also shown at ESMO 2021 that the addition of pembrolizumab to neoadjuvant therapy and continuation in the adjuvant setting significantly and in a meaningful way improved event-free survival, with an increase in the cure rate and a reduction of recurrences from 24% in the placebo group to 16%, which accounts to a hazard ratio of 0.63. At San Antonio, we presented additional data. We presented pre-specified sensitivity and subgroup analysis just to assess the robustness and consistency of the primary event-free survival data in a bit more detail. In the primary analysis, we calculated event-free survival and events as local progression that precluded definitive surgery, distant progression, local or distant recurrence, death from any cause, or positive margins at the last surgery. And the purpose of the sensitivity analysis, which were all pre-specified, was to look at some of the censoring rules which were possibly affected by how often disease recurrence was assessed and by missing disease assessments or by starting a new anti-cancer therapy. We also looked at different definitions for events, for example, positive margin at last surgery, how that was counted, or a second primary cancer. All 5 sensitivity analyses were pre-planned, and all 5 sensitivity analyses were very much consistent with the primary EFS results, showing that the data for the EFS benefit were really highly robust. We also, on these sensitivity analyses, for example, include the impact of adjuvant therapy. A small percentage of patients received adjuvant capecitabine outside clinical trial, and even taking that into consideration, the results were not affected or not changed. We also looked a bit more in detail in patients with nodal involvement as compared to patients without lymph node involvement. And this was based on the initial observation that the path CR rates were, the delta, the additional benefit was higher in patients with nodal disease as in patients without lymph node involvement. But when we look at event-free survival, the patients derive the same relative benefit, whether they are node positive or node negative. The hazard ratio for node positive patients was 0.65. For node negative patients was still 0.556. And the difference in 3 event-free survival rates was about 7% in patients with node negative disease. Finally, we presented an update on some of the safety data, which basically confirmed the results we have previously reported, demonstrated that the combination of pembrolizumab and neoadjuvant chemotherapy is safe and in keeping with the safety experience what we know for immune checkpoint inhibitors. So in summary, we have shown that there's a statistically significant and clinically meaningful improvement in event-free survival with the addition of pembrolizumab to neoadjuvant chemotherapy and continuation of pembrolizumab in the adjuvant setting. We were able to show that the treatment benefit with pembrolizumab on event-free survival is robust across different subgroups, and also that each of the sensitivity analyses showed a very similar result. As such, we feel that the addition of pembrolizumab to neoadjuvant chemotherapy is a new standard of care in patients with high-risk stage 2 or stage 3 early triple-negative breast cancer. We are substantially improving the outcome of patients, short-term and long-term, reducing recurrences by nearly 40%, including distant recurrences reduced by 39%. Pembrolizumab has already been approved by the FDA in the US and is now a new standard of care for patients with stage 2 and stage 3 triple-negative breast cancer. And I would anticipate that the drug is also approved in other parts around the world and will become the standard of care in these countries as well.